The second speaker of this session is Rich from Roma, and the title of his talk is Chemical Synthetic Biology. Yeah.
So I will tell you about chemical synthetic biology, and I hope to be able to explain to you what this concept is about. Let me say that for me, synthetic biology is challenging and interesting because I convert this idea of making alternative life form of life structure into the question, why this and not that? Why nature did things in a certain way and not in another one? Why DNA has ribose instead of glucose? Why does RNA has uracil instead of thymine? And when you ask this question, then synthetic biology can offer you the alternative of making DNA with glucose or RNA with thymine and see what the alternative form of life or structure of life can do. And from this comparison, you can learn, we believe, a few things about nature. So let me show you some of these examples. No. Oh. Yeah. And let me face the first question by asking, for example, why our proteins and not a different set of proteins? This question has a rationale in, in the well-known calculation that you can do with your students, considering that you have 20 different amino acids. When you have a chain of, let's say, 60 residue, and you want to ask the question, in how many different ways can you realize this chain of 60, you end up with this 20 times 20 times 20, with a number which, according to young Einstein, corresponds to the number of atoms in all universe. Now, in our world, we have a few billions proteins, an easy calculation, very many, but of course the ratio between the theoretical number and the actual number is something staggering. It's something that corresponds, this ratio, to the ratio existing between the space of the known universe and the space occupied by a single hydrogen atom. Or if you want a more earthly and poetic rendering, if you take the whole Sahara sand, the ratio between the whole Sahara sand and one single grain of sand corresponds to the ratio between the number of theoretically possible protein and the actualized ones in our world. And this simple calculation elicits then a question: how have these few proteins of our world be selected out? Do they have something special like solubility, stability, folding? And that's why have been selected out and chosen. In other words, are the proteins of our life the only one that could be formed and gave origin to life by a deterministic, by obligatory step? Only those could be formed, or the opposite view, the contingency view, are they the product of chance? They have been chosen by the contingent conditions at the origin of life, and then life and we are the product of chance. You know, this is one of the big philosophical questions attending the whole life science, the ratio, the controversy between determinism and contingency. Is the origin of life an obligatory pathway? It had to happen the way it happened, or is it due to chance? Now, in the case of proteins, let's try to approach this kind of philosophical question in a very experimental way. Let's make in the lab a library of proteins which do not exist on nature, the ones that we call the never-born proteins, as evolution had never had the time or the will to make them. And then let's compare these never-born proteins with the extant protein and see whether then they differ in properties like stability, folding, solubility. Folding being the main characteristic feature of our functional globular protein. And this slide is not very clear, but I have a number of bright co-workers like Cristiano Chiarabelli, Davide Lucrezia, Fabrizio, and Anna and Alessio, who are, most of them are here having obtained several grants. In my time, this didn't happen. Now, how do we do this library of never-born protein? Is a technique which is well known in the molecular biology literature. Is called phage display. For details, I remind you to the several posters we have. But essentially what you do is the following. You ask a company to make for you a large library of random DNA, so constructed randomly by computer. And if you do by random a sequence of DNA 150 residue long, the probability of hitting an extant DNA is 1 over this 10 to the 70, is almost 0. So once you have this library of random DNA, you insert one by one. There is, of course, a statistical procedure to do that in a virus. And these produce a protein, which corresponds to this non-extant DNA, so it's non-extant protein. And you give this phage to an E. coli, which produce them, and then you end up with a library of a protein attached to phage with a sequence which is completely random, and except for a tripeptide, PRG, proline, arginine, glycine, which we put there because this tripeptide is specifically recognized by a protease, thrombin. And the rationale for doing that is to discover which structure are folded, as, of course, let me first go here, if you have a protein which is rigidly folded, a protease cannot bite inside and will resist proteolytic digestion, whereas if you have an unfolded proteins, then protease can attack and cleave and hydrolyze very rapidly. So of these, we produce, Cristiano and company produce a library of about 1 billion clones. Of course, they couldn't work with all of them, despite the long time for PhD work. And Cristiano has chosen only a few clones taken by random, and about 73 of them have been studied in terms of folding and stability. And the results are as follow. I summarize by saying that at the beginning, the question, how many of this structure will be folded? And some of our colleagues were answering, you don't find not one, because, to find a folded, randomly created protein, you may need 10 to the 30. Well, in fact, the contrary is true, that about 20% of these never-born protein are resisting the attack of thrombin, and with our definition, which is limited but in first approximation real, 20% of these proteins are folded. It's a kind of binomial distribution. A lot of them are rapidly destroyed. And here you see the computation of some of these never-born protein computed structure with the Rosetta method, and with the limit of this method, but still the best in the market. And you see here the characteristic feature of our already known protein with helical turn and beta turn. I mean, if I would have told you that they are never born, I would think that they are normal proteins. And in some case, in a couple of them, we have like these. Let's skip the details. You can do spectroscopy, for example, fluorescence. This structure has only one tryptophan exposed to water, so denaturing and the natural spectrum is equal in terms of the emission fluorescence to the native one. And you have another one, not that we have made many, in which one of the tryptophan is a little buried, so that the denaturation brings about a slight red sheaf. Most important are perhaps this circular dichroic measurement. For those of you who are expert in protein folding and protein structure, they will know that this CD spectrum is characteristic for a folded structure, and this peak here gives you information, a kind of quantitative or semi-quantitative information about the content of helix. So you denature the protein with guanidinium chloride, here stops, then you take away guanidinium chloride, and the structure refold as it was, as showing a kind of reversible denaturation as in our textbook. So that here a first tentative conclusion could be that completely random de novo protein can be stable, water-soluble, folded. They have, so that our proteins are not unique in that. Actually, one could arrive at the temptation of saying, then our protein are perhaps then the product of contingency, as they have nothing special from the structural thermodynamic point of view. You may not agree with this kind of strong philosophical statement, but this is an indication how experimental work can, tackles the problem of philosophical question in the field. Philosophy, we heard very little in this meeting until now. And of course, if you do never-born proteins, the messenger that produce this protein are also never-born. And here, Davide, who should sit here. No, yes. You can ask him all possible detail, but what he did is, together with Chiarabelli and others, is to find a criterion for testing the stability of RNA structure by using this RNA which is active on single strand. And you see here that by increasing the stability of the RNA, the resistance the sensibility to this enzyme decrease, and playing with literature, with temperature, does the opposite. So you can do also study, and David was able to found a clone of RNA that was stable at 60 degrees, so a thermostable RNA, not as a product of evolution, but as a product of random synthesis, which is perhaps of some we are doing now other libraries, and I may tell you later why. But let me go quickly to the second example of chemical synthetic biology, making proteins which not by the use of molecular biology. Again, a good team of co-worker, a little younger than I, and partly like Pasquale are here. And the idea is to do fragment condensation. You have fragment 1 and 2, and you join them in these. And so from these, let's say 10 mer, you make 20 mer, and you join the 20 mer, you make the 40 mer, and you use that. And a test case has been done with a series of peptides long 10, 4 of them condensed with other 4, so you get 16 by the Merrifield method. And then we leave the contingency of environmental pressure work. Those are all insoluble products, and we decide to discard them, simulating a kind of environmental pressure. They precipitate, they're out of business. We get these 320 mer, and these 20 mers are condensed with other randomly chose 20 mer, obtaining a library of 40 mers, and letting the environmental condition work. We found that only 2 of them were slightly soluble under the denaturing conditions, and adding 4 amino acids to one of them, you got a never-born protein with a stable folding, stable up to a certain temperature. You can predict the structure, etc. If you, instead of the Merrifield, you want to do the kind of prebiotic game, you can do that. I don't have the time to give you detail, but the idea is to use simple peptides, which are so simple that they can be the product of prebiotic peptide synthesis, to do the job of fragment condensation. This may sound strange and odd, but if you look in the literature, you find that the group of Yufeng Zhao and others have already found that certain simple peptide can cleave the peptide bond. So if there is a catalyst that cleaves, the same catalyst can do the condensation by changing the conditions. And we have studied, for example, the simple reaction catalyzed by this seryl histidine, and you get an incredibly good yield. Without going into detail, simply notice that the yield of synthesis of this product catalyzed by the simple peptide is very high, comparable to chymotrypsin. Chymotrypsin does it in few minutes. With this peptide, you need a long time, but in prebiotic time is not so important. So again, it's an indication that by chemical mean you can produce a functional macromolecule, which is still a mystery in the origin of life. Let me go quickly to the last part. Again, the question, why this and not that? Why are our living modern cells so complicated, so complex? If you look at the simple beast like that, a few micron across, very simple indeed, but if you zoom inside with the zoom of a biochemist, you find this incredible complexity. This is part of the E. coli. Each point is a product, each line is a reaction, each reaction is catalyzed by an enzyme, which is and each product is the substrate of another reaction, is an incredible complexity. Yes, but the early cells, when life started, could not have been so complex. So let's look for the notion of the minimal cell. Let's try to make, by synthetic biology, oh, this is horrible. I don't have the time to change the, but this minimal cell contains the minimal and sufficient number of components to be defined as a life. Here is a kind of definition of a life. We can come back. What we do is to use liposomes, vesicle made by lipid, into which, with our technique, we became expert in over the last 10 years. So we introduce extant genes and enzyme, but trying to do the minimal amount of them so as to obtain the minimal living cell, and for that we use liposomes. There are these vesicles that when you look under the microscope, you have already the impression of something like life. Of course, they are simple bubbles full of water, and they're just pretending. But those are capable of self-replication. This is our old work that started in 92. You don't see here with this article on Nature, and went on with a series of papers on vesicle. Here the reproduction of vesicle, they are caught in the act. And while they are replicating, you can put something inside, for example, Q-beta replicase, which is an enzyme that makes copies of a RNA template. So you have a self-replicating shell, and inside a self-replicating genetic material, a kind of caricature of cell life. Of course, is no life, because with the progress of generation, since nobody does for you again the enzyme and the RNA, there is what we call death by dilution. Now, a series of groups following our early step in the early 90s are in this field of making cells, and the exercise now to put inside liposome a kit capable of expressing proteins. Presently, we are working with the system developed by Ueda in Tokyo, consisting of 37 enzymes, but then if you count the ribosomes and the tRNAs, and there is a total of 90 macromolecular components, and we were able to put these components, each of the concentration of each of them is known, which for a chemist is very, and here is the expression of green fluorescent protein inside this vesicle. And we addressed in this field a number of questions. One was, how small in terms of size can a cell be, and be capable of be a cell, for example, expressing protein. And we could scale down to 100 nanometer radius and still have protein expression. Now, we are trying now to do self-replication of this minimal cell, which implies the self-replication of all the components of the pure system, including the ribosome. The question, how many genes are necessary to do that, to arrive at a minimal DNA protein cell, we calculate around 50, but we have to see with experimental work whether this ambitious number is possible. And this is part, of course, of this general field of, which is central to synthetic biology, synthesizing life in the laboratory. The idea that life is an emergent property, namely something that arises by putting parts together, parts which are not living per se. You put them together and the new property arises, which we call life. Whether this is true or not, it still has to be completely proven, but it's certainly one implicit assumption and ambition of synthetic biology. So, in conclusion, I hope to have given you an example of synthetic biology, which we call chemical synthetic biology, as in all this exercise we don't play, we don't interfere, we don't make violence to the genome of living organism, with, you know, this question, why this and not that, which I like very much. And actually, I give you, as last slide, the word of very famous molecular biologist by name George Bernard Shaw, who says, you see things as they are, and you say, why? But I dream things that never were, and I say, why not? Which is, in a way, another definition of synthetic biology. Thank you for your attention.
So now the session is open for questions and comments.
I guess my question was whether if you took those sequences that were unfolded and you put them through Rosetta, did you find that they were random sequences as well?
It's a big question, and we are struggling with it, because with the Rosetta method, you always get a kind of more stable structure. And in principle, you don't know whether this is completely folded or not. So what we are doing now is to combine the Rosetta method with dynamic measurement, because the unfolded structure should be more flexible, and that's the current state of the... But, you know, in the literature, this very question is never addressed, and it's a very good one. I'm thankful for this question.
Are there any other questions?
Oh, my friend Yomo.
Just a simple question that
I don't believe you.
Binomial distribution against the foldability or resistant to the thrombin digestion, right, at the beginning, before the selection. Some of them are quite, you said that they hold, and then some are unfold. Are there any relationship between that foldability to the solubility? I'm just afraid that some of the folded in your definition might relate it to the aggregation or somehow.
It's certainly a problem. You know, these never-born proteins are easy to build on Blackboard, and actually it's easy, relatively easy to make a library. What is very difficult, what is the bottleneck, is the isolation and purification, because of course this is not described in the literature, and aggregation is indeed a problem, and we are struggling with it. And whether the the discarded one are soluble, we have never studied the discarded one. We have concentrated on these folded clones, so must be done still. Sorry, Ron.
So, is there any other questions or comments? Okay, so if not, let's thank Pierluigi again. Yeah.